Thank you so much. Thank you, Axel. It's always a pleasure to be here at JGI. I love the talks, so it's really nice to be the last speaker because I have the benefit of having heard all of the amazing talks. So today I'm going to share with you something that I've been excited about in the last few years, that is to marry cell biology with metabolism. And the pictures that you see here are all the beautiful aspects of plant cells that I think are really exciting to get into. So let's get into it. So a few years ago, I started an initiative called the Plant Cell Atlas, along with Ken Birdbaum and at NYU. And, well, I'll let you know when we should kill the lights. Sorry. Well, you guys are doing a great job. Thank you. Yeah, so this initiative was started with Ken Birdbaum at NYU and David Earhart at Carnegie Science. And we now actually have quite a few people, around 90 people leading this initiative, along with Ben Cole at JGI. And it's a community effort to understand and engineer plants at the single cell level so that ultimately we may be able to actually engineer whole plants. And I love using this example as kind of our poster child of the opportunities we have with single cell approaches. And it comes from work from Ben, actually. And so this is something that has to do with plant defense against pathogens. So each year, up to 40% of our crops lost through the pathogens. And it's over $200 billion a year of loss. And until now, we've been really thinking about plant defense as something that happens at the whole plant level. So we talk about plants that are resistant or susceptible to pathogens. But using single-cell approaches, Ben and Gerda Croker's group at UC Davis have actually found that even within the same plant, within the same leaf even, you can actually have cells that are more susceptible and cells that are more resistant. This, to me, is kind of groundbreaking because this really defies the way we've been thinking about engineering resistant plants. So even in a single organ, you may actually have diversity of the same cell type, diversity of cells that can actually be more resistant, and we don't understand where that variation comes from, okay? But we may actually have a handle on it using single cell approaches. So we're very excited about it, and the community is growing. So we have over 2,500 scientists around the world who are members of this initiative, with close to 300 members actively interacting in Slack. And we have about 90 committee members that are doing different types of activities and services to train and engage scientists around the world. And I just want to plug that we're going to have a Gordon conference next summer in Maine. And it's going to be preceded by a GRS, which is actually a Gordon Conference pre-meeting just for students and postdocs. So I hope a lot of people here might be interested in going there. And so now I want to come back to the idea of metabolic cell biology. So we know that metabolism happens at all scales of the plant or organism. Here I'm using plants as an example. And I'm particularly interested in the scale at the subcellular level, okay? And I'm really interested in this for many reasons, which I'll go into. Cellular biology or cell biology is not a typical way that people study metabolism. So far, people have been looking at metabolism largely from organ level or in vitro biochemistry, with cell-free extracts, or genetics, actually, has been really useful. But I think cell biology has a really important place in understanding, helping us understand metabolism. This is just a beautiful image of a single cell that has different parts of the organelles lit up with markers. It's work from Amanda Koning in Zhangping Hu's lab at MSU. And I just really love images like that because it gives you a sense of how metabolism might actually happen in real time in space. Unfortunately, metabolism is really not well studied at the cellular level. So if you look at how many enzymes of plant metabolism have been localized at the single cell or subcellular level, it's not very many, and most of it comes from Arabidopsis. the yellow here are the, what is yellow? I think yellow is like non-enzymes, and the green is enzymes, okay? So there are about 1,000 enzymes that have been examined at the subcellular level. And there's about 150 for rice, 70 for maize, and then like only 4 in sorghum in the literature right now, okay? And very little for all other bioenergy crops. For example, the really important photosynthesis pathway in plants, or especially in C4 plants, essentially none of the enzymes other than rubisco has been really characterized very well at the subcellular level. So we went out, so before talking about what we did, I want to say that knowing subcellular compartmentalization is really important for improving metabolic efficiency. So this just shows you a few examples, like, you can increase local concentration of certain metabolites of interest. You can actually reduce the production of things that you don't want. You can also protect, the metabolites from damage from environmental stress, for example. And in fact, compartmentalization has been used very successfully in increasing the production of certain metabolites, and metabolic engineering has really leveraged this fact. Even in plants, we have examples of several examples of increasing the production of interesting metabolites by several-fold. So a few years ago, funded by DOE's BER and in large collaboration with JGI, actually, in synthesizing DNA, we set out to characterize the subcellular localization of sorghum enzymes. Okay? I want to thank especially Ian Blaby and Ritesh Mevalal at JGI for synthesizing hundreds of these genes for us. Okay, so now we tried a bunch of different approaches. I'm going to take a moment to take a sip because I'm going to tell you some bad news. So we were really excited about using carbon nanotubes to transform plants. We also tried very hard to transform leaves of sorghum transiently. And then we also tried this aquatic plant, Agaricea densa, that only has 2 cell layers in the leaf, and it was pretty easy to transform using bombardment. And also we tried sorghum protoplasts. And most of the stuff didn't work, actually. I would say for the first like 75% of this grant funding period, we just had negative results. And then the Agaricea densa, and especially sorghum protoplasts, were the ones that really came to the rescue. And so we ended up doing most of our characterizations in the protoplasts, actually, of sorghum. And now we have a very beautiful system that works well, except that my postdoc who's leading this effort, Purva Karia, will tell you there's some seasonality associated to the protoplast working. And I think it's the same for all labs that are trying the protoplast right now. Somehow it stops working, and then magically it starts working again, when nobody can really figure out what the factors are. It's kind of curious. If we had more labs working on this, I think we might actually solve that problem, but it's an interesting problem. So how did we do? All right, we actually tried to cover all of the enzymes in the metabolic pathways of sorghum bicolor, and we chose 408 enzymes to characterize across 168 pathways. And we localized mainly using sorghum protoplasts, and we were able to localize close to 300, which I think is a 72% success rate over the 5 years or so that we were working on this project. And what's more important is 73-fold increase in subcellular localization of sorghum enzymes out there. Right now, putting the work together into a paper and a database that will be accessible to everyone. Okay, and so this next set of slides just shows you some of the results that we found, and I want to sort of end this part of the talk with a few interesting insights and potentially novel compartments, but it's too early to tell probably. But anyway, this bar graph shows you that we covered all the domains of metabolism. The green bars represent the portion of the enzymes that we covered in each domain, and the gray bars show the enzymes that we still need to do if we were to cover 100% of the metabolic space. And of the 300 or so enzymes that we characterize subcellularly, half of them are actually, let's go back. Half of them are in the cytosol. Some may think this is not as interesting, but I love to think and look deeper into to see if there are any like sub-compartments of the cytosol itself. That would be quite interesting. The rest of the half are kind of interesting. They, we have expected things like chloroplast and ER, mitochondria, and so on, but we have quite a lot that are just in puncta, okay? And also quite a few that are just in vesicles. We still have to, do a better job with co-localizing with peroxisome, but some of these puncta and vesicles could potentially represent new compartments. And so we're kind of excited about that. We do have to do a lot more due diligence, but it's an interesting early result. So now, just to show you the proof of concept of we were able to localize enzymes where we were supposed to localize them. Now we can maybe kill the light. I'm just showing you the C4 photosynthesis of mesophyll cell and the bundle sheath. So carbonic anhydrase, as expected, should be in the cytosol of mesophyll, and you can see that it's in the mesophyll. Can you see it okay? Should we kill more lights? Yeah, let's just try to kill all of the lights just for the next couple of slides. Yeah, is okay? Okay, that's pretty good. And then the NADP malate dehydrogenase, which is supposed to be in the chloroplast of the mesophyll, we see it in the chloroplast, and some of you may see that it's like nicely in sort of maybe stroma. NADP malate enzyme, which is supposed to be in the chloroplast of the bundle sheath, is also found in the chloroplast. But it's also found in the cytosol a little bit too, actually. We don't know whether this is because of overexpression, or maybe this is actually a mesophyll cell that's trying to express a bundle sheath enzyme. We don't know yet. We know from single cell sequencing work that we can get bundle sheets and mesophyll cells pretty well. And so we have to actually use markers to distinguish them, and we're in the process of doing the co-localization for bundle sheath and mesophyll. Now, I want to tell you that there's some interesting things we found. So we looked at all of the enzymes for the Calvin cycle in sorghum, and shown in orange are the enzymes that we couldn't localize, actually. And an example of that is Rubisco, small subunit. Here you see them going into the chloroplast, but that's only because we used the first 80 amino acids. So we had to truncate the C-terminal end in order to see it going into the chloroplast. So there is some post-transcriptional regulation going on with Rubisco's small subunit, and we hypothesize that the rest of these orange enzymes are actually the same. And I don't think this is known yet in the field. It looks like a lot of these Calvin cycle enzymes are post-transcriptionally regulated at the C-terminus for their localization into the chloroplast. The blue enzymes we were able to localize into the chloroplast, as you can see. As some of you might notice, though, some of them actually go into this punctae. So right now we're trying to see first if they're phase-separated lipid droplets, or if they actually all go to the same puncta or not. So Rubisco has been shown to go into these biocondensates in clammy, shown by Martin Jonicka's group. And so in our hands it doesn't, but it's partly because we're only using the first 80 amino acids. So it would be very interesting to see if we can see additional compartments of of Calvin cycle enzymes in sorghum and in other plants too, for that matter. Another really interesting that we found is having to do with shikimate biosynthesis, which is really important in making not only amino acids, but a lot of important secondary metabolites in plants. So shikimate pathway, of course, is known to be operating in the chloroplast. But what we found is this. So when we actually localized all of the enzymes of the shikimate biosynthesis pathway, we found everything to be localized in the chloroplast except this. So this is shikimate dehydrogenase, which catalyzes two consecutive reactions, but it was not in the chloroplast. And we tried this multiple times with multiple tags, and it's definitely not in the chloroplast. It's in the cytosol. So then we went back to the literature, and in fact, we learned that this actually has been observed a long time ago. Roy Jensen actually called this dual pathway hypothesis for shikimate biosynthesis in 1989, where he said this pathway may occur both in the chloroplast and, oh, that's exciting. At least it's not happening to the slides. Maybe that's Roy. He's like, yes. Yeah, and so the pathway here I'm showing you, basically there's been evidence that these enzymes are found in multiple places in the cell. So the green dots are the enzymes that are found in the chloroplast only. The brown are the ones that have been found in the chloroplast found in the cytosol only. And the blue are the ones that have evidence that's found in the cytosol genetically. So the brown is the one that's been found in the cytosol biochemically. So you can see that the same enzyme, the shikimate dehydrogenase, where is it? Right there, has evidence from biochemistry and genetics that it's in the cytosol. Okay? So we, for the first time, are showing, using live imaging, that we see it in the live cell in the cytosol. So it's very possible that this pathway may actually shuttle this intermediate into the cytosol and bring it back in. And so, of course, there will be a whole new lines of experimentation that can be done to find the transporters. What does this mean in terms of metabolic engineering? It's a very important compound and pathway to engineer. And so this was another exciting sort of finding. The third example I'm going to give you is chloroplast targeting, which again, we think is very well characterized and known, right? So there are these transit peptides and signal peptides that take proteins into the chloroplast. And they're always in the N-terminus, and so when you want to tag chloroplast localized enzymes, you always do the C-terminus tag. And so Purva found this really interesting case where these 4 enzymes went to the chloroplast, okay? But they were not tagged at the C-terminus. They were tagged at the N-terminus, okay? And when she tagged them at the C-terminus, they no longer went to the chloroplast. They were in the cytosol, okay? So there is potentially some non-canonical chloroplast targeting signal that's not in the N-terminus, which is potentially interesting, okay? So I'm going to summarize this part of the talk by saying that we localized close to 300 enzymes in sorghum to their subcellular compartment. And we found this would be a really good resource, I think, for a lot of different things. We're particularly interested in using it as a training data set to really help annotate subcellular localization patterns. But I think it will, as I've given you some examples, open doors to looking at certain pathways and certain engineering approaches. So I'm going to just give you one vignette to kind of end the talk. I don't know, I think I have maybe another 10 minutes or so, to give you an example of an interesting subcellular localization that turns out to have very interesting biology, okay? This sort of extends the concept of, localization to function paradigm. Okay, so the pathway that I want to talk about is rhamnose biosynthesis pathway. Rhamnose is a sugar that's made by a lot of different organisms, not only plants, but also invertebrates and bacteria, to make the cell wall for plants and mucilage and flavonoids, as well as glycoproteins in various organisms. Okay? Bacteria and protozoa use this sugar to make lipopolysaccharides, and invertebrates use it to make cuticle. It's a simple pathway that basically converts glucose, UDP glucose to UDP rhamnose, and it's basically 3 enzymatic steps: dehydration, epimerization, and then reduction. And it's carried out in plants by a single enzyme. There's a helper enzyme, which I'm not going to talk about today. Its enzyme is called rhamnose-1. And in Arabidopsis, there's 2 more homologs, rhamnose 2 and 3. But anyway, rhamnose-1, catalyzes all these 3 steps, and it's actually a fused protein. It's an example of what, forget the previous speaker, example of nature's way of doing metabolic shuttling. Okay, so they fuse these 2 domains that exist as separate enzymes in bacteria into one plant, called binding domain 1 and binding domain 2. So binding domain 1 actually catalyzes the first reaction, and then binding domain 2 catalyzes the last two reactions, okay? So, so this has been well characterized, and in the literature, it's known to, the enzyme is known to localize to this puncta, okay? But it's not described any further. The people just say it's in puncta. And so because our lab started working on biocondensates a few years ago, we wanted to know if this biocondensate or potential biocondensate of this enzyme may have to do with its function at all. Okay? So there are two ways that this kind of physical biocondensation could influence activity. Okay, one is it could inhibit reaction by preventing substrates from getting in. Okay, another way is to enhance the reaction by channeling the substrates. In the literature, there are two evidences that support either of these hypotheses, basically. Okay, and so we wanted to sort of go deeper into that. First, we actually asked, okay, where is this working? Where can we see a phenotype? And when you knock out Rhamnose-1, it's been known that you can actually get very, very interesting cell wall and developmental defect in the flowers, especially in petals. So when you get rid of, when you get rid of Rhamnose-1, instead of getting this very beautiful cone-like shape of petal cells, you get this flatter cells, and the secondary wall formation is, instead of being radial, it's more parallel. And so we first asked, where is this body starting to form, or when can we see it? And so we looked at different stages of flower development, especially when the petal's expanding. And I think we are going to need to kill the lights completely if possible. We basically see these bodies for, initially, the enzyme is found in the cytosol like this, in early petals, stage, I think this is stage 11. Okay, so there's a small petal. When there's like expansion, a huge expansion going on, we start to see the enzyme localizing into these bodies. And then when the petal senesces, the body goes away. And so, yeah, we can turn the lights back on. So then we wanted to know, okay, what is the function of this body, the rhamnose body? And so we took advantage of yeast not making rhamnose, and we expressed Rhamnose-1 in yeast to ask if we can start to examine the structure-function relationship, especially with respect to the body formation. So what we did was, using AlphaFold and modeling, we identified a residue that may be involved in dimerization of the protein. This is one A161 right there. And we removed that, we replaced that residue to lysine, I believe. And we got rid of the body formation, as you can see in yeast beautifully. So wild type makes this really nice bodies in yeast, and then when you replace that residue, we no longer got the bodies. And so then what happens to that phenotype I showed you in the petal? So again, here it shows you that these bodies go away in the mutant, and we are, let's see, if you have the wild type petal here, and then this is a Rhamnose-1 knockout. If you add the wild type back, you recover that petal phenotype and the cell wall phenotype. When you put back the version that cannot make the body, we are not able to recover that phenotype. And also, we saw something similar in the mucilage production in the seed, as well as the overall growth of the plant. This is just showing you the rosette. So that mutation that prevents the body formation actually is no longer able to function. And we also showed that, just to make sure that we can do this with the catalytic residue as well, we mutated the catalytic residue, which is the glycine 16 here, to alanine, and that one is able to make the rhamnose body, but it's no longer catalytically active. And when we look to see if rhamnose is produced, we see that this graph shows you the amount of rhamnosome, or we call it rhamnosome, the rhamnose body formation. So wild type makes it, the mutant that doesn't make the body does not make it in yeast, and then in the one that's mutated for the catalytic site still makes the body, but both the body defect mutant and the catalytic mutant no longer can make rhamnose. Some of you might be wondering, well, what if that residue that you mutated that gets rid of the body formation is actually kind of interfering with the catalytic site? Because it is kind of a little bit close, even though it's on the other side of the enzyme. We then went back to AlphaFold and found another patch of the protein on the other side that will really potentially get rid of the body formation, because it's also predicted to be involved in oligomerization of this protein. And when we got rid of all 4 of these residues, we got very clean phenotype of not having the body, okay? And this quadruple mutant is actually, interestingly, quite defective in making the rhamnose, okay? And one of the mutants, though, is interestingly makes more of the intermediate. We don't know exactly what this means, but I think there's something that may have to do with channeling that metabolite through. And so we're going to look deeper into that. I think we're going to have to look at the actual structure. So we're trying to do cryo-EM on this. Okay, I'm almost done. I want to say that I started by saying this pathway is conserved in bacteria, and these are some of the animals, and these are some of the organisms that make rhamnose. And we asked, okay, so if the rhamnosome enzyme in plants are in these biocondensates, and the biocondensates seem to be important for making rhamnose, is this body found in bacteria? And so we looked at E. coli, C. elegans, and a couple of other, Arabidopsis thaliana is one that we started out with. And we looked at it also in Trichomonas vaginalis. I believe it's the protozoa. And they're all found, they all make these bodies to a certain extent. And we're actually very interested now looking at this in Bacillus subtilis, because that's a really important model for biofilm formation. So this is kind of our model about how this rhamnosome might be involved in bringing the enzyme together to facilitate the synthesis of rhamnose. And this is really interesting now in terms of understanding channeling of metabolites and metabolon function, because there aren't really a good model for metabolons, because they're all very, very complex, lots of enzymes involved. But this single enzyme metabolon could be a really interesting model for looking at the structure function of metabolons. And so I want to end by thanking the people that worked on this. Purva Karia is a postdoc who led that really courageous sorghum metabolic enzyme location study that we call Sorghum Metabolic Atlas Project. She was helped by Maxine and Ava. Ava is now a grad student at NYU. And Sterling Field is the postdoc who led the rhamnosome study. So I'd like to thank you for your attention and take any questions if you have any.
Thank you, Sue. That was really exciting. Mysteriously, we are back on time despite all of the AV issues, so we have to have time for a few questions.
Okay.
Do we have any? Yeah, back there.
Do you think there's some sort of allosteric activation between the two domains such that the enzymes don't work as well when they're monomers?
We haven't, maybe. We haven't tested to see if we can break it apart and then put them together to see how they would work and whether they would actually still form the biocondensate. So that could be really interesting to test, actually. But we are testing that in E. coli, where the two domains are actually in separate enzymes. And so the picture that I showed you with E. coli, that's just looking at just the first enzyme in the pathway, the BD1. So yeah, we don't know if there may be interesting interactions going on with the sort of allosteric interactions and the two domains that are physically fused, if that's what you're asking.
Thank you. Very thought-provoking.
Very exciting. My question is about enzymes that are part of pathways that are traditionally known to localize in cytosol and chloroplast, like the pentose pathway or other things. So within the list of enzymes that you were able to cover, do you have any sense of how many you were able to cover both of those? And the results, were they as predicted?
Yeah, I think most of the localizations were in single. I should actually include that figure. I don't think I have it here. Most of the localizations were in single compartments, but there were quite a few that were in dual, like cytosol chloroplast was the main dual localization. And I have to double check to see if they validated what's known in the literature. But yeah, I think they do.
Well, actually, it wasn't about a single enzyme dual localizing. There are two copies, that one of which...
Oh, isozymes.
Yeah. Yes.
Yeah, we've done lots of isozyme characterizations, and some do validate, but the isozymes is really interesting case. There are quite a few that are not expected. So we don't really know how to handle that. Is that like a new variant of the pathway that wasn't really characterized before, or is it like that those are giving us mislocalization because that isozyme is actually not in the leaf? So that's one of the kind of limitations of doing everything in leaf protoplast cells, because you're kind of like limited to that one cell type, or largely two cell types, I think. The bundle sheet and the mesophyll. I think it would be really great to be able to do this ultimately in like stable transgenics, or maybe like try to get protoplasts from other cell types. I know it's kind of not easy to do from roots, so I don't know. But isozymes are very interesting. I think it would be a really interesting way of looking at function diversification through localization diversification. And we're looking into that.
Thank you.
Okay, I had a question actually. So for the compartmentalization study in sorghum, that is really cool. So if I understand correctly, you look at the different genes one by one. Mm-hmm. Can you target two at the same time, for example, GFP and some other fluorescent protein? Mm-hmm. Yeah. And this might be particularly interesting with respect to the currently unknown subcompartments that you had, like where you can, so you might in a targeted way take those that seem to go to the same general compartment and see if they actually co-localize. Have you tried that at all?
We are trying that. That's how we're doing the co-localization. So we're doing co-localization using known organelle markers. Some of them are dyes, but most of them are proteins. And so that takes a huge amount of effort because the efficiency, kind of transformation efficiency goes down. So we can get 80% efficiency if we transform very small peptides. So I think that the new direction might be to try to identify markers that are really like small tags. We have some, but some of them are like full-length proteins. And so efficiency is about 80% if we just do small peptides, but it goes down to 20% if we do full-length proteins. So co-localization, brings down, combinatorially the efficiency even further. So it's a little bit challenging. We could potentially add like two, genes in the same construct, but again, like the more length you increase, the lower the transformation efficiencies too. Yeah. So we're kind of battling that kind of challenge. Yeah. But yes, we are doing co-localization, and we now want to do co-localization. I think that's where you're getting at, co-localization with enzymes, different enzymes of the same pathway, like that. The Calvin cycle I showed you, where we see puncta of different Calvin cycle enzymes, we want to see if they're all going to the same site or same puncta.
Yeah, exactly. And you might even be able to quantify it pretty precisely by measuring the different fluorescent signals.
Yep, we're doing that right now.
Great.
Yep.
Sorry. Can I ask a question?
Yep.
This is about the transit peptide, or the possibility of a new kind of transit peptide. Is it the idea is that that the transit peptide is not necessarily located at the N terminus,
Where is the speaker?
Oh, sorry, over here.
Okay. Yeah.
Yeah, or is it possible that they're actually talking about a completely different pathway for getting into the plastic?
Ooh. I think maybe it could be either of those options, right? It could be that maybe there's a helper protein that is N-terminal tagged, and this is just like this protein is just like a cargo, which means that there's a new pathway maybe, but it's still like not there. It doesn't mean that there's a different kind of signal peptide. So it could be that, or it could be that there's a different like signal peptide that's not at the N-terminus. I have no idea, so we're going to have to keep digging into that. We looked into the literature, and there's only one study from Arabidopsis that reported 10 proteins that were localized to the chloroplast without, with the N-terminal tagging. So, whether this is artifact or new biology is something we have to really dig deeper into. But one possibility is like that there's a facilitator for transport that we don't know about, or that there's something going on in the middle of the protein or at the C-terminus. Oh, sorry, yeah. But when we just take the C-terminus part, it doesn't go into the chloroplast. So I don't know, it's like, I don't know what's going on. Yes. Yeah, that could be possible, yeah.
Okay, I don't see any other questions. If you have one, last call. Wave really hard. Oh, okay. So, thank you again, Sue. Thank you again to all speakers in the final session.